And can everybody see like the full presenter mode?
Yes, it looks very nice.
And my pointer?
Yes. Although if you point at that blood meal, it will disappear with red on red.
Yeah.
Well, we've made it to our penultimate class. Next week we'll have one talk, a makeup talk due to illness, and the second half will be a debrief. All right, I'm gonna mute myself and become an interloper.
Alrighty, so I'm seeing 11:30. You ready to go, Benjamin?
Yep.
All right. So hello, everybody. It's my pleasure to introduce Benjamin Phipps. So Benjamin earned his bachelor's degree from the University of North Texas in 2019, double majoring in biology and Spanish. Before coming to Georgia, he's now a second-year student in Dr. Michael Strand's lab, working on the interaction between malaria parasites, its mosquito host, and the microbiota of that mosquito. So go ahead and take it away, Benjamin.
All right. Well, thank you for that introduction, Sam. And so today I'll be presenting my research plan for my dissertation project, which will focus on the roles of mosquito neurohormones and gut microbiota in oogenesis and plasmodium or malaria parasite development. And we're interested in mosquitoes because they are the most dangerous animal on the planet, to humans anyway. They kill more people than people do annually. And they do this because female mosquitoes, most female mosquitoes in most species have to take a blood meal in order to produce eggs. And they often take multiple blood meals within their lifetime to support producing multiple clutches of eggs. And in doing that, they can acquire and then transmit various deadly pathogens of vertebrates. And the most destructive of these is the malaria parasite, which is responsible for over half of the deaths caused by mosquitoes. And it's caused by parasites in the genus Plasmodium, which are transmitted by mosquitoes in the genus Anopheles. And 5 species, in fact, humans, with P. falciparum being the most deadly. But this is also a larger ecosystem problem because there are many different species that specialize on different vertebrates, from mammals to reptiles. And these parasites have a complex life cycle that involves a mosquito and a vertebrate host. So when a mosquito ingests parasites in a blood meal from a vertebrate host, the parasite must undergo a complex series of stages before it can become infective to the next host. And human immunity to malaria is poorly understood, and this complicates the design of an effective vaccine. And therefore, prevention strategies focus on vector control. And with the advent of technologies such as gene drives for suppressing vector populations, it's critical to find targets that can be exploited to suppress mosquito populations and or parasite transmission. And one of the primary targets for this is, or several of the most important targets are within this process called vitellogenesis, which is the synthesis of yolk and packaging of that yolk into oocytes. And the primary pathways involved in this process in mosquitoes are the insulin signaling and target of rapamycin or TOR pathways. And the way these pathways are activated is nutritional signals from the blood meal stimulate the brain to secrete these neuropeptides, which include ovary estrogenic hormone and the insulin-like peptides or ILPs, and we're especially interested in ILPs 3 and 4. And so this nutritional signal from the blood meal, only the blood meal can activate this vitellogenic process. And so these peptides stimulate the ovaries to produce a steroid hormone called ecdysone, which in turn triggers expression of vitellogenin, which is the major yolk precursor protein, by this nutrient storage tissue in insects called the fat body. And then vitellogenin is taken up by oocytes via receptor-mediated endocytosis. And so primarily these endocrine processes have been worked out in Aedes aegypti, which is sort of the model mosquito, but it's unknown to what extent this is conserved in Anopheles mosquitoes, which are the really important mosquitoes because they transmit malaria. And certainly homologs for these genes have been identified in Anopheles mosquitoes, but to what extent they're functionally conserved is unknown. And another unknown is precisely what signals after the blood meal trigger this neuropeptide release. And there's some evidence to suggest that microbial metabolites are the stimulant, because firstly, all mosquitoes host communities of bacteria and fungi in their digestive tract, and they require, under standard rearing conditions, gut microbes to develop to adults. And this is in part because these microbes are important for proper regulation of insulin signaling, and we've seen that insulin signaling is dysregulated in azenic larvae, and also that egg production is impaired in azenic female adults, which suggests that insulin and reproductive signaling is also impaired in the absence of microbiota. Azenic means basically germ-free. And these reproductive processes are thought to be tightly linked to the life cycle of Plasmodium, because these physiological changes in the mosquito are triggered by the blood meal, which if the blood meal was taken from an infected host, also contains the parasite. And so it's been demonstrated that uh, Plasmodium manipulates insulin signaling, and also that if these metallogenic processes are disrupted by ecdysone receptor knockdown, this impairs Plasmodium development. And Plasmodium also has this connection to the microbiota because antibiotic treatment of mosquitoes increases susceptibility to Plasmodium. And so people have been trying to work out why that is. And we've seen a lot of literature about the direct interactions. So these microbes are known to secrete compounds that are toxic to the parasite, but to what extent indirect interactions affect Plasmodium survival by maybe changing the uh, by impacting the physiology of the mosquito in a way um, that makes it more or less able to support the parasite. That's not so well understood. So I have a few questions about all this that I intend to answer in my dissertation research. How do OEH and ILPs impact oogenesis in Anopheles stevensii, which is an Indian malaria vector? And how do gut microbes impact oogenesis in Anopheles stevensii? And finally, how do reproductive hormones and gut microbes affect Plasmodium development in the mosquito? So to answer my first question, if OEH and ILP3 are involved in, they play important roles in vitellogenesis like they do in Aedes aegypti, we um, think that most likely they would be upregulated after a blood meal. And so to answer whether they are, and also to answer whether they're primarily expressed in the brain, I'll conduct RT-qPCR in the head, midgut, and ovaries for to measure transcriptional expression of these hormones in blood-fed versus sugar-fed mosquitoes. And then to probe the functions of these hormones, I'll first ablate the source of these hormones by decapitating females. And believe it or not, in many insects, the individuals that are decapitated can survive for several days. And so, and this has been done in our lab with mosquitoes. And so far, I have seen that blood-fed mosquitoes that are decapitated fail to produce eggs. However, I plan to rescue egg production by injecting decapitated females with recombinant OEH and ILP3. And so far, I've seen down here that that injecting decapitated mosquitoes with ILP3 essentially completely rescues oogenesis as compared to intact mosquitoes. And I'll further probe the roles of these hormones in stimulating egg production by knocking down their putative receptors using RNAi, and I expect that in these females, this will abrogate egg production. And then finally, to determine whether these hormones um, trigger vitellogenesis by inducing synthesis of ecdysone, I'll do this in vitro destroyed assay that has been developed in our lab where we expose explanted ovaries from non-blood fed females with recombinant OEH and ILP3, and then we can quantify ecdysone production with an ELISA. And then for my next question of how do gut microbes impact oogenesis in Anopheles stevensii, I'll similarly probe the functions of the gut microbes by essentially ablating them by rearing azenic or germ-free females. And we've done this in our lab and have seen that we can rear these to adulthood and get them to take a blood meal and everything. And so I'll feed these azenic females a blood meal and count the eggs laid. And we anticipate that if these microbes are essential for egg production, these azenic females will fail to produce eggs. And then to determine whether these microbes are influencing egg production by influencing digestion of the blood meal, since the nutrients from the blood meal are required to make eggs, or by directly influencing the reproductive endocrinology. I'll first to use an enzyme activity assay to quantify protease activity in the midgut, as well as quantification of neutral lipids in the midgut and the fat body to determine whether lipids are being shuttled. And I'll compare those between azenic and conventional females. And then also perform RT-qPCR to measure OEH and ILP3 expression in azenic versus conventional females. And then to determine whether specific microbial species are key players in egg production, I will inoculate azenic mosquitoes with individual microbial species that we've previously identified by sequencing and characterizing microbial community composition, and assess egg production in these notobiotic females, or females with defined microbial communities. And we expect that most likely some microbial species will influence egg production and lead to greater egg production capacity than others. And then finally, how do reproductive hormones and gut microbes affect plasmodium development in the mosquito? First, to identify the roles of reproductive hormones in plasmodium development, I will infect OEH and ILP3 receptor knockdown females with Plasmodium berhei, which is a murine malaria parasite, and assess parasite survival and proliferation. And I'll also do this in these infection experiments in azenic females to identify the roles of gut microbes in plasmodium development, and further to dissect the interplay between the parasite development and gut microbiota and endocrine signaling, and how the gut microbiota and the parasite may have competing effects on hormonal signaling and expression, I'll conduct RT-qPCR to assess expression of these hormones in azenic versus conventional, infected versus uninfected females. And then finally, to determine whether the same microbial species that affect egg production similarly affect plasmodium development, all infect these notobiotic females with the parasite and assess the parasite development. And I wanted to briefly mention this on the rigor and reproducibility. Previous experiments have used antibiotic-treated mosquitoes and done these infection experiments, but this poses a concern for reproducibility because of the ubiquity of antibiotic-resistant bacteria as well as eukaryotes that aren't affected. And this means that there can be differing results between regions because of the differential abundance of certain antibiotic-resistant species. And so I intend to eliminate the background signal from different resistant species by rearing mosquitoes that are truly germ-free. And just briefly, this is our process: is we surface sterilize eggs and rear up these larvae in water containing E. coli K12, which is ampicillin resistant. And then we surface sterilize these pupae and treat the adults with ampicillin, which wipes out the E. coli, and we end up with azenic females. And this can be verified by homogenizing mosquitoes and plating them to see if there's any growth, as well as PCR for using universal bacterial and fungal primers. And so this work will identify targets for future malaria and vector control strategies, and potentially also identify microbial species that can be exploited for disease control. And so I'd like to thank the members of the Strand Lab for providing support in my research. And I'd also like to thank my committee and my mentors for providing feedback on my presentation. And thank you for your attention. And I'll take any questions.
Great. Thanks so much, Ben. That was a great talk. I have put the survey in the link in the chat, and we're going to open up questions. I will get it started, Ben. You were saying in your first question that decapitated females don't lay the eggs, so they don't have that like hormone, right?
Yeah.
Because you're thinking it's brain. Have you thought, has it been shown that injecting them recapitulates vitellogenesis? I'm just thinking about like a simple, you could do like a simple like head swap. So I was wondering if you had thought about doing that at all, just to see if like the head swap led to anything before. I mean, I'm assuming injecting them isn't as simple as just switching heads, but yeah.
I haven't thought about doing a head swap. You mean like swapping heads between?
Like you're taking decapitated females, so they don't have the hormone, but what happens if you just put a head of a different female back on it? to
Oh yeah.
recapitulate
Mhm.
vitellogenesis?
Yeah. We've done where we kind of where we homogenize heads and we inject the extracts.
Okay.
Um, and we've seen that rescues egg
Okay.
production, but we haven't done like a head swap. But yeah, we've done something similar with the
Okay.
head
Cool.
homogenous. Yeah.
Cool. Alrighty. And then you guys can put in the chat if you guys have other questions for Ben, or just unmute yourself. All right. If there's no other questions, the link is in the chat again, and it is live. Thanks again, Ben.
Mhm.
That was great.
I know everyone's still filling out the survey, but we want to make sure to keep some time, or maintain the time. So while you're filling that out, I'm gonna have Mike pull up his slides and get ready to share the screen. Okay. All right. So it's my pleasure today to be able to introduce Michael Francis. He got his bachelor's in biochemistry at the State University of New York and the Environmental Science and Forestry College in Syracuse. After that, he did his master's at the University of Georgia, and he is now a second year in Kaizhang Yi's lab. Today, he'll be talking to us about the gene-environment interactions of fish, oil, and triglyceride levels in humans. And with that, I'll let Mike go ahead and take it away.
Thanks, Derek. Can everyone hear me okay? Great. So as Derek said, I'm going to talk about gene-environment interactions of humans taking fish oil supplements and how this affects their triglyceride levels. I'm going to play a video. Hopefully it works. Go. Let food be thy medicine, and medicine be thy food. These words were spoken by Hippocrates, father of modern medicine, around 400 BC. Today we understand the basic premise well enough. For example, eating a fresh salad is known to be healthier than eating a box of cookies. But the details become more complicated when it comes to choosing the right dietary supplements or maximizing weight loss with low carb versus low fat diets. How can we know which food is the right medicine for each and every person around the world? Okay, so that's sort of a million miles up overview of the topic. So I'll dig in a little deeper. So one of the ways we can answer these questions is through genome-wide association studies. So I'm sure most of you know, but in case you don't, in a GWAS, you can take two groups of people. You can take a case group and a control group. Let's say the cases have cancer and the controls don't. What you can do is take their genomic information and compare it and find significant differences between the DNA content of these two groups and figure out if there's a genetic component to the disease. We can look a little closer and see that you can use a genotyping panel and compare the same variant in both these groups and create what's known as a Manhattan plot. This is sort of one of the main visualization tools of a GWAS. And what we see here is the negative log of the p-value. So essentially the higher the dots go, the more significant the association is. And we can see very easily the loci that are significant differences between the two groups.
And when we're thinking about GWAS, we want to think about the common disease, common variant hypothesis. So up here in the upper left hand corner of the graph, we have highly penetrant Mendelian mutations. Something like if you have one variant, you're going to get this disease. Like cystic fibrosis is like a very powerful mutation, and it has a very large effect. But for things that I study, like metabolic diseases, obesity, type 2 diabetes, Alzheimer's disease as well, what we're going to be thinking about is common variants working in a group that all have small effects. So there's no like obesity gene, right? But what you have, typically there's like 80 variants that are used in a sort of a panel, and you can create a polygenic risk score and look at how all these variants appear together in a person and can cause a different phenotype. So I'll switch to talking about continuous traits. I know I was talking about cancer, which is a binary trait, but I'll talk about continuous traits. The only difference really is that for a binary trait, you're actually looking at a logistic regression, but since I'm doing a continuous trait, I'll just explain it this way. So we can think about a standard GWAS formula as being like a linear regression, where Y is the phenotype. So for example, triglyceride levels can range from maybe, I don't know, 3 to like 7 millimoles per liter, I want to say. And for this G term, so the betas are the effect sizes, and for G, it'll be like the non-major allele dosage. So for example, if you have SNP, you know, RSXXX, if the major genotype is TT, and then the heterozygote is TC, and then the homozygous recessive is CC, we would just code this as 0, 1, and 2, according to how many C alleles are there. And then the covariates, so usually things like sex, age. I'll talk about the specific covariates I used a little bit later, but this is sort of just a general way to understand it. So for interaction, what we do is just add one more dimension of complexity to that. And so what we can do is take out one of the covariates and actually call it E as an environmental exposure. So in my experiment, we use fish oil supplementation as sort of a binary 0-1 environmental exposure. Do you supplement with fish oil or don't you? And then you can add this G by E term, which is going to give you the interaction. So this is like a statistical regression concept. I'm not really going to explain it mathematically, but the way to understand it is just how these two terms are interacting, right? So there's things that the genotype is doing, there's things that the presence or absence of the exposure is doing, and then there's also a third thing, which is how they work together. And this sort of the sum of their parts is the whole is greater than the sum of their parts sort of thing, right? So what is interaction and why does it matter? So DNA does not work alone to produce phenotypes. You know, you can think of like a nature versus nurture type of question. And many common diseases have an environmental component which alters the effects of genetic variants. So we can use gene by environment interactions to better understand biological processes leading to disease, to identify subjects for whom risk factors are the most relevant. So if you're looking strictly at DNA, you might miss the important factors that are causing people to get sick. And we can also improve the accuracy of our models, right? If there's a whole dimension missing from the model, then it's not going to be accurate. So what I'm looking at are gene diet interactions, which is a specific type of gene environment interactions. It's basically the same thing in practice. It's just where E is a dietary factor. So this is like a nutritional genomics subset of this gene environment interactions. So for example, when looking at lipid trait phenotypes, things like low density lipoprotein, cholesterol, HDL, or triglycerides, gene diet interaction between omega-3 fatty acid supplementation and genetic variants can help explain things like missing heritability in these traits. So for example, you know, my mom and my dad both have high LDL, but I don't. What's happening there, right? So there's not just a strict heritability happening here. And then also a really important thing in the previous literature of omega-3 supplementation studies is this heterogeneity of response. So, you know, you have a whole group of people, and we're giving them all fish oil, and even when their DNA seems to be similar, the response to the supplementation is different. So this is another way that we can explain that phenomena. So I want to talk about the results that I got in my most recent paper, which is in revision right now in PLOS Genetics. So I'll just talk about what we did. So the background is that we're looking at dyslipidemia, so imbalances in LDL, HDL, and triglycerides. The reason we care is because this is a common predictive factor in humans for cardiovascular disease and type 2 diabetes. And also using dietary supplements instead of pharmaceuticals to manage this condition can produce comparable benefits with fewer side effects. So that's really interesting. And the long chain omega-3s, EPA, and DHAs supplied by fish oil supplements are often shown to be beneficial in the literature for hyperlipemics, so people with too high lipid levels. So the study set I used was UK Biobank, and this is the largest open human health research database currently. The total data set is 500,000 people aged between 40 and 69 years old. And this data was taken between 2006 and 2010 in the UK. And it's 88% white British, and that's important because we use only one homogeneous race group. And it's just to avoid population stratification, which is a really important consideration in GWAS experiments. So you want to reduce your confounding variables, and one of the things you can do is just take one racial group. So this data set is actually good for that. And our exposure, like I said, was fish oil supplementation. So when you're doing G by E studies, accurate or unbiased environmental exposure is super crucial. And one good thing about this study is we actually have a subset of participants that gave their fish oil supplementation status twice. So for example, they were asked in 2006, do you supplement fish oil regularly? And then again, in a follow-up assessment 5 years later, do you supplement fish oil regularly? So it's not perfect. Like a lot of nutritional studies, it's not perfect, but it does help us get a more accurate picture of if these people are supplementing regularly. And of course, the cost to this is a decreased sample size, but considering the data set is so large to begin with, we were okay with that. So we did our original QC, and then we took people who only did the follow-up 24-hour recalls survey, so the second nutritional survey. And what we ended up with was a little bit more than 100,000 participants in the final data set. And so for covariates, we use things like common GWAS covariates, sex, age, socioeconomic status, BMI. One thing we also add to this experiment was the number of servings of oily fish they ate. So for example, even if you're not taking fish oil, but you are eating, 5 servings of salmon every week, right? That matters. So we want to know that. And then the first 10 principal components, and then fish oil status, yes, yes. In other words, yes at the first time point, yes at the second time point, or no, no, no at both time points is our interaction term. As far as the software we used, I used Plink 2 mainly for quality control and for file conversions and just sort of, mundane tasks like that. The actual GWAS analysis was done with QuickTest, which is really accurate gene by environment interaction analyses. And then we used a software called Metal to do 2 degree of freedom tests, which I'm not going to talk about because it takes a minute to explain, but also our meta-analyses mainly. So it was our meta-analysis software, and this really cool tool called FUMA that you can sort of apply at the end, which checks if your loci are new compared to all the known GWAS catalog data. So it's a 3-stage GWAS design. I'll just give you the overview. You do a discovery phase. So this was UK Biobank. So we found about 26,000 significant associations in the discovery. And then we attempted to replicate these in a study set called ERIC. So totally different people. And what we found was about 2,000 replicated studies, replicated associations rather. And after our meta-analysis, we used the genome-wide association significance level commonly used 5 × 10⁻⁸. And what we found are 4 loci of interest. And I'll just talk about this one because this was the only locus that didn't have a significant main effect. So it seemed like a strictly interaction locus for triglycerides. So I'm going to focus on that locus here. So that's this RS 112803755. It's our main SNP of interest. So what I'm showing here is a locus zoom regional Manhattan plot. So similar to the Manhattan plot I showed in the beginning of the presentation. This is a locus zoom plot. So just this one section of chromosome 13 we're looking at. And so here is our significant lead SNP of interest, this RS112, et cetera. And the cool thing about a locus zoom plot is it'll give you the SNPs that are in linkage disequilibrium with this SNP. So what we find is it's highly enriched in this cluster right here. So this GJA, GJB, GJB cluster, sorry, GJA, GJB gene cluster, which encodes for connexin genes. The nice thing is that connexins have been previously shown in mouse studies to actually change their gene expression upon exposure to omega-3s. So we were in the right universe here, right? At least there's some biological context that we can place these results in. And so this is pretty cool. I'll just go through these one by one. So the minor allele is significantly associated with decreasing triglycerides in individuals taking fish oil, but with increased triglycerides without fish oil. Okay, so let's look at this first graph. So what we're showing here is the effect significance of the SNP, like the main effects, and that's super low, not significant. Here's the genome-wide significance threshold. And what we find is that the interaction term is highly significant. I'll skip over the joint for now, but one cool thing is that we can actually stratify our population into 2 groups, like those who don't take fish oil and those who do. And you can find that this SNP has a higher main effect, which is really the interaction effect that you're measuring at that point in the group that takes fish oil. So this is very robust. This is exactly what we were looking for. And so this graph, so on the left, we have people who don't take fish oil, and on the right, we have people who do take fish oil. So the major allele, the major genotype AA is a little bit, a tiny bit higher in those who do take fish oil, but then we see a divergence here, right? For those who don't take fish oil, their triglycerides go up on average when you get the heterozygote, but for the heterozygote who do take fish oil, it's actually a decrease in triglycerides, right? So it's a little hard to explain in the limited amount of time, but I hope you're getting the main picture that basically there's a divergence of groups depending on whether they took fish oil supplementation, right? And as far as rigor and reproducibility, our findings were robust, as I just showed. There's mechanistic plausibility of our main findings, and in all GWAS studies, you publish your summary statistics to public servers so everyone can see them. I'm not just making these numbers up. And it's important to note that the results of GWAS tests don't necessarily reveal the causal variance. What they do is provide a framework for experimental validation in future studies. And with that, I'd like to thank my lab and my mentors, Derek and Yibing. And that's it. Thank you very much.
So, is anyone have questions here?
Hey, Mike, I have a question.
Sure.
Awesome. I was wondering if you could talk a little bit more about the missing heritability that this, I guess what my question would be, do you think that how much of an impact does fish oil, I guess the environmental fish oil supplementation factor into the missing heritability that I guess how much does the fish oil supplementation, do you think, affect the actual the phenotype of, or play into the missing heritability that people, I guess, are finding with sorry, I forgot explicitly what you said in the introduction, but I think you get my question.
Yeah. You want me to talk about the missing heritability more?
Yeah.
So like in terms of exact effect size, it's hard to really say. If you think about the pipeline of sort of experimental discovery, right? Like I'm way over here on the sort of like just initial, kind of here's like a million SNPs, right? And I'm saying maybe I can narrow it down for you to like, a small handful. And then eventually what will happen is that future labs will see this paper and say, okay, let's experimentally test this SNP maybe in like a cell culture, right? And then eventually maybe it'll get to like a mouse study, human study, right? So eventually many steps down the line, we'll get to a human study where we can more accurately quantify the actual effect size. So, I don't really want to say like, okay, we determined it's like point whatever, because, but the significance portion of it is notable, but the actual effect sizes of a GWAS, especially since this is the first GWAS in the first population. So remember, this is just for this population as well, right? So, you could get another, even American Caucasians, not to mention every other race under the sun, right? And you could get completely different results. So, so we're very early in the discovery phase of this. So I hope that answers your question.
Yeah, definitely. I was initially thinking about your data set and how you had but I imagine your data set might not be as robust to include like whole family structures. But yeah, okay, thank you.
It doesn't. Yeah, some human data sets like biobank data is set up in like trios. UK Biobank is not set up that way.
I have a question.
Sure.
When you were talking about the data, you said that you used the PC axes 1 through 10, right? What made you decide to use that? Is that a common thing with human GWAS or, yeah?
Usually it's just the convention of these type of studies to use the first 10, yeah. As you know, with PCs, it's sort of like an exponential decay of impact as you keep going. So we actually did look at these, and it definitely starts to level off around 10. So we thought that was fine to follow the convention in that sense.
Cool. Yeah, I'm just curious since I'm using a different organism, different system. So.
Does anyone has more questions? Okay, I already put the questionnaire link on through the chat. Please fill them out.
Thanks, everybody.
Hey y'all, thanks for coming to our penultimate class. We have one more class meeting next week. We will have the first half of that class as a makeup talk due to illness, and the second half will be sort of a debrief. So do spend some time mentally thinking about things that worked well this semester, things that didn't work well this semester, because I'll be transferring over learning lessons, what do we want to keep or not keep as we go forward to a new instructor in the next semester. Especially for some of the senior students, you've experienced the older style and the newer style. For some of the newer students, you may only know COVID style. So any class can always be better. So I welcome that discussion next week. Otherwise, I will close the meeting today. Do remember to fill out your questionnaires. I will turn them off at 2 o'clock. All right. Thanks all. Bye-bye.